DNA, or deoxyribonucleic acid, is the code that is responsible for having engineered every cell in our bodies. It is the code that is responsible for the development of every living thing on our planet. The combination of adenine, cytosine, guanine, and thymine, or ACGT, are the four bases that make up our unique genetic code, or signature DNA. It's no secret that forensic scientists have been using DNA in blood, skin, saliva, and crime scenes to identify perpetrators. The unique signature that DNA leaves behind is what makes it ideal to forensically mark security instruments like currency passports and driver's licenses. Methodologies from the Human Genome Project have established the standards for conducting DNA research and analysis. It is truly the global standard today for all forensic analytical work from the lab and now also in the field. Researchers from one company, Applied DNA Sciences, have discovered the genetic code from plants that provide the perfect vehicle to secure products, and now it can help with the recovery and identification of stolen cash. From fine art to pharmaceuticals to even automotive parts, Applied DNA Sciences is the only company in the world to use the genetic codes derived from plants to mark products and authenticate them. Over 1 billion products around the world are now using signature DNA to protect their products from counterfeiting, diversion, forgeries, and even theft. This approach has already been independently validated by the Department of Energy at the United States National Laboratories. Here's how it works. Large, complex plant-derived DNA sequences are encrypted, stabilized, and then used to individually mark the ink for each cache box. When a cache box is attacked, the ink is instantly dispersed inside the box with our signature DNA marker. Once the cache is stained with the DNA marked ink, it cannot be removed, replicated, re-engineered, or digitally copied. When a cache box is recovered with the stained notes, the first step of analysis is to view the note under ultraviolet light. Like our company logo, which is blue, you will immediately see that the Applied DNA stained notes will glow bright blue under UV light. When you find a bright blue stained note, the second step is to submit the sample for DNA analysis. The note should be placed inside a forensic sample collection envelope, following the standard operating protocols established by Applied DNA. Each sample that we receive into our labs will be handled with the utmost security. We will process the sample information into our secure database and will prepare the stain note for analysis. Each note is carefully prepared by extracting the ink from the note. We will take multiple samples from each note, purify the DNA, and prepare the DNA for extraction. Each purified DNA sample is placed inside a PCR machine, which amplifies the DNA code that we have extracted from the stained note. Once the PCR is completed, we will compile a certificate of authenticity to verify that the DNA code was present and authenticated. Some companies suggest that their products are like DNA or have similar forensic features like DNA, but they simply cannot be and are not as secure. Signature DNA is the only true forensic DNA marker. It is what makes every cash in transit box unique and it is the ultimate in crime prevention.